species. And being in my lab, she also leveraged a little bit of citizen science data on breeding birds as part of her research. She also quickly decided to switch to being a PhD student, and she got additional funding from teaching, from being an instructor of record for a course, and also TAing, and this year as a global change fellow with the Southeast Climate Adaptation Science Center. Anyway, Murry is a super accomplished public scientist, right? Since 2020, she's done like over 35 public science and educational activities like talks and workshops and podcasts, media interviews. She has like this awesome bird banding workshop that's tailored to different age groups, like with plush toys for little kids and real birds for older kids. So for those of you that know Murry, well, or at least I'll say how I know Murry, which is to be a fairly quiet and very concise person. Like I haven't found her to be the type who just talks for the sake of talking. But when she has something to say, I guarantee you that it's something important and that she won't hold back from saying it. And so I wanted to highlight a couple of things that Murray's really been saying. And one thing that she's had to say is that there weren't enough children's books about nature and science that featured kids that look like her. And so she's been fixing that. Right, she's the author of a children's book series with Christy Activano books with Little Brown Young Readers, is the publisher. And her upcoming book, Sparrow Loves Birds, features a 5-year-old Black girl learning how to identify common backyard birds. And it's the first of a series of her Sparrow Loves Nature series that's themed around exploring and appreciating urban and suburban wildlife through the eyes of a minority main character. And another thing that Murry has had to say is that field safety looks different for marginalized biologists, right? In her first field season, which her fieldwork is in rural North Carolina, her first season was in the spring of 2020. And if you recall, there was like racism experienced by Christian Cooper while birding in Central Park. There was the murder of George Floyd, and it was also the start of the COVID-19 pandemic. And there was Murry in the field working in a barn on a farm that was owned by a really fantastic fellow and managed by a really great guy, supportive guy. But the next door neighbors, not so welcoming, and various homes with Confederate flags and not so welcoming political messages all along the way. So Murry became very outspoken about field safety from her perspective as a Black female biologist and birder. She had me buy car door magnets so that people could see from a distance that she was affiliated with NC State. And she raised awareness about this issue around the college. And then she co-founded Field Inclusive with another grad student, Lauren Farr. And Field Inclusive is a nonprofit to raise awareness about social field safety issues and offer field safety training and resources and provide financial assistance to marginalized biologists. Anyway, and I want to congratulate Murry on all that work and thank her for it. So Murry earned her bachelor's degree from the Wildlife, Fisheries, and Aquaculture program at Mississippi State University, that's in Starksville, Mississippi. And now they want her back ASAP. They've offered her a job as an assistant professor. And anyway, I'm sure that Murry is going to go on to do many great things that are worth doing. So without further delay, I'm going to hand things over to Murry to present about her research related to artificial light at night and how it affects reproduction in birds. Go ahead, Murry.
Let me unmute myself and share the screen. And I need to reshare. Okay. Thumbs up if you can see that. Yes, okay, great. So thank you for that awesome introduction. And without further ado, I'm just going to jump into my presentation. So my research focuses a lot on photo period, clutch size, everything related to light and birds. And my first research question for my first chapter, Change in Photo Period Best Describes Pattern of Clutch Size in Eastern Bluebirds. The question is, what determines avian clutch size? And the title is kind of spoilery, but let's explore how we got there at least. So there is this ubiquitous trend with clutch size, and that is one, across latitudes, clutch size increases as you move towards the poles. And additionally, within latitudes, clutch size decreases over the breeding season. So we see this trend like pretty much everywhere in like pretty much every species, especially in passerines. And so one of the biggest questions that researchers have been looking at for a long time is, what's the explanation for this trend? So there's been various explanations across different years. For the latitude, why does clutch size increase as you move towards the poles? Some researchers have said that, well, it's because parents have more access to food in northern latitudes because the days are longer in the north, so they have more time to forage, more resources to feed their chicks with. Some other researchers say that it's really a trade-off between predator avoidance and reproductive success. However, that doesn't quite fully explain the latitudinal trend. That's more of a regional trend, since predation rates are going to vary and don't vary in the same pattern as clutch size does. And some other researchers further have said that seasonality is what determines resource abundance, as well as competition during the breeding season. For example, competition between resident species and migratory species who have come into the area and also want the resources. As for the seasonal aspect, the main theory is that early breeders lay larger clutches. And some reasons for that is that they might have access to higher quantity and quality of food because they got there first. Access to better quality mates because they got there first, and access to higher quality nesting sites. The true definition of early bird gets the worm here. However, these all explain only just one portion of the trends that we're seeing. So what I've tried to do is put all of these latitudinal and seasonal trends together and explain it with just one explanation. So my explanation is looking at photoperiod and the biological clock. So pretty much all living organisms have a biological clock, including humans. If you've ever woken up before your alarm, or if you get like hungry at the same time every day, that's your internal clock telling you what time it is. Biological clocks are typically driven by photoperiod, both daily photoperiod and seasonal photoperiod. And this is really important because these cycles synchronize animals' behavior and physiology with their environment, making sure they're getting the resources that are available at the time, telling them when to migrate for better resources or better environmental conditions, when to hibernate, etc. So it makes sense that photoperiod would be the main factor. As an abiotic factor, it's ubiquitous across the globe, just like these clutch size trends. So it makes sense that photoperiod might be the one. And so building out the case for photoperiod specifically in birds, birds have many neuroendocrine functions that activate and regulate in response to light. So for birds, light not only is detected through their eyes, but also through complex systems involving the pineal gland, the pituitary gland, hypothalamus, all these different organs and hormones that they secrete. So it makes sense that birds are highly sensitive to changes in light, no matter how small the changes in light are. And photic input regulates clock genes and these physiological pathways through signaling secretion levels of various hormones, including hormones that regulate like the initiation and termination of the egg laying period. So that's really important. So in order to kind of test this, I wanted to see if changing photoperiod best describes as the only explanation the latitudinal and seasonal patterns of clutch size. So I had this large data set from the Cornell Lab of Ornithology's NestWatch and subsetted it based on Eastern bluebird data. There was a huge data set with over 58,000 records, and it spanned from the years 2000 to 2019 across many latitudes and over a wide range of breeding seasons. is also really popular. Bluebirds are really popular with backyard birders and observers, and so there's just like a ton of data on them. And with that data set, I was able to use the SunCalc package in program R to calculate both the day length and the change in day length, the change in photo period, which is just like how many minutes longer or shorter was today than yesterday. And I tested 3 linear models with this information. So the clutch size is determined by latitude and lay day. Clutch size is determined by day length, or clutch size is determined by the change in photo period. And the results were that clutch size, that the clutch size and photo period model is the most robust out of all of these. It has the highest R² value, lowest AIC, and highest log likelihood. And so this lends evidence towards the theory that change in photo period is the thing that most accurately describes clutch size. So what does that mean? Just implications from this first chapter is that researchers can like disregard latitude and date as just human constructs. You know, birds don't know that it's July 20th, 11 a.m. They just go by the seasonality that they detect. And this might also inform that the traits under selection is not just the number of eggs in a clutch. It's you have to take into account all of the physiological mechanisms that control the laying sequence. And so jumping from there into the next chapter, this is taking what we know about photoperiod and how important it is for reproduction and development and saying, wait a minute, but there's a whole lot of artificial light at night that's kind of interfering with that natural photoperiod cycle. So the question here is, are nestlings experiencing any sublethal effects from anthropogenic stressors? We know that anthropogenic stressors impact birds and a whole bunch of wildlife. This is a very important study, especially like just within the whole urban ecology realm, because about 80% of the world's population is estimated to live in an urban environment. So it's not just humans, but a whole lot of wildlife, a whole lot of birds that are going to be experiencing various anthropogenic stressors. And like I said, the one that I focused on for this study is artificial light at night, also sometimes referred as light pollution, and I'll probably be calling it ALAN throughout this presentation. But so artificial light at night comes from street lights, buildings, advertisements like stadium lights, decorative lights, lights on a bridge, just any artificial light that is on throughout the night. And sky glow, you might think that ALAN only is an issue in the cities where the light source is. However, artificial lights do create sky glow, which affects all environments from like an urban to a rural gradient. And sky glow is just the scattering of light particles throughout the atmosphere so that the light particles obscure the natural light of the stars. In fact, over 1/3 of the global population cannot see the Milky Way. And just in North America, that number is as high as 80% of people not having star visibility. And light pollution is expected to continue to increase, as urbanization increases, from 2 to 10% per year by some estimates. So this is a huge issue that's going to have a huge ecological impact no matter the particular ecosystem that it's in. And narrowing it down to how ALAN specifically impacts birds, there's a lot of studies showing that ALAN changes their migration or causes some disoriented navigation, which could include effects like grounding, which you see in this kind of upper left picture, grounding from birds being tired, or maybe they hit something and are injured now, and grounding puts birds in a very vulnerable state. Light pollution also causes building collisions, which there's like over 1 billion building collisions per year, and that can be, all of those first 3 points can be lethal to birds. In addition, studies have seen that Allen leads to earlier dawn song, extended foraging periods in birds, including earlier in the morning and or later in the evening, as well as earlier gonad production, earlier arrival at nesting sites, and earlier reproduction. So there's been a lot of studies done on this community and population level effects. But what I'm focusing on is specifically the physiological effects. Advance. There we go. The physiological effects on nestlings. So obviously, if the parents are experiencing these effects, parents affect how the chicks are raised. In particular, if the parents are having an extended foraging period under artificial light, then that might impact the amount of nutrition that the nestlings are getting. So one of the things that I'm especially looking at is the signs of an urban phenotype. So a phenotype just looking at a specific behavior, morphology, and physiology that is seen in populations or have been experimentally induced with artificial light. And this is kind of what is known. So nestlings raised in Allen sometimes experience increased begging. They might be awake throughout the night, begging for more food throughout the night. They have higher metabolic stress markers, such as high cortisol levels or lowered immune function, which indicates stress. They also end up with lower body mass, both as they grow, when they fledge, or even when they first hatch in artificial light. They have low body mass, sometimes an overall lower body condition, and artificial light often leads to lower brood success overall. So this is everything that is known, these kind of characteristics of an urban phenotype. But the question is, what are the internal mechanisms that are leading to this stress or this low body mass? So the high metabolic stress markers does mean that they are stressed, but why does that result in behavioral and physical differences? And are these physical differences that we're seeing more of sublethal effects? So one of the things that I decided to look at in my research is the role of glucose and how glucose might affect this urban phenotype that we're seeing. So first of all, altricial nestlings in regular conditions always have a high energy demand during development because they go from egg size to basically adult size in a matter of weeks. And so when they demand energy, they get energy from their food and store it as glycogen in the liver. And when they need more energy to build their bodies to become adult size, they use a process called gluconeogenesis, which is just taking the glycogen out of the liver and using it to feed their energetic demand. And this kind of cycle is the typical role of glucose under like the best circumstances. It leads to nice, strong chicks and ready to become fledglings. However, what happens if you add in stress into that system, a lot of stress? Like usual, chicks will attempt to store extra glucose as glycogen in the liver and use gluconeogenesis to satisfy their energetic demand. However, if they're like super stressed and there's not enough glycogen to fuel gluconeogenesis, they might turn second to breaking down proteins in their bodies, using amino acids to do gluconeogenesis. And if they're using up all the glycogen, if they're using proteins that should be going towards building, their muscles and their feathers, are they getting enough to build their body? Are they getting enough to satisfy this stressed out fight or flight response? It's a little bit unclear how that pathway will resolve, but it does most likely lead to these scrawnier chicks with this urban stressed out phenotype. So in order to actually test this, I conducted a barn swallow artificial light at night field experiment. This barn is in Snow Camp, North Carolina, just about an hour away from Raleigh. It's a rural area, and I chose a rural area so that I'm able to isolate the source of light pollution to only the artificial lights that I put up. I also chose barn swallows because one is really convenient that they nest in these wooden structures, so I can like hang up lights rather easily. Also, I thought it was fitting to use a global species that is experiencing population declines. However, those declines have not been attributed to Allen, more so to like pesticide use on farms. And so I did this experiment for 3 years, and I tested the hypotheses that Allen is an environmental stressor that causes sublethal effects in nestlings. And the second part, that these nestlings exposed to Allen will have higher blood glucose because they're so stressed, and slower growth because they're using some of the proteins that should go towards their body towards fueling the stress response. So a general methods overview, I randomly selected nests in this barn to control an artificial light at night treatments. I placed lights above all of the nests, but only turned the treatments actually turned them on from sunrise to sunset every day. The kind of lights I used are 3000 Kelvin or warm white LED lights with a minimal lux of 1.6. These levels match typical commercial and residential lighting, and the intensity of the lights matches low-level street lighting, so trying to create like a realistic situation. The control nests were measured at an average of 0.0 lux, which I added a little asterisk here just to acknowledge that you can't truly have a zero measurements of lux. It's just the power of the light meter that I had. The full moon can be like as high as 0.3 lux. So, but during this experiment, I recorded metrics of nest activity, morphological and physical development, and for statistics, general statistics, I use linear mixed models with the random effect of nest, because we know that parents are going to have an influence on how the chicks are raised and try to control for that genetic factor, and as well as repeated measures of the chick. These are some pictures of how my experiment was kind of set up. I would go at night periodically to take my measurements, make sure everything was working, lights were turning on and off when they were supposed to. The top photo, you can see this is my first iteration of the setup. I was using Christmas lights and using tin foil to block out all of the bulbs except the one that was like directly over the nest. However, we had a huge issue with predation with that setup. The rat snake you see there was able to travel along the Christmas lights and eat my research. So we had to try to change the types of lights to these flatter strips LED lights. And again, I blacked out most of the lights to prevent spillage, only had like one single section above the nest lit. And I did make sure that both strands of lights were at the same color, temperature, and intensity. For data collection, I started off right from the start with nest monitoring, wanted to record the lay date of the first egg, when the eggs hatch, what the clutch size was, the brood size, as well as keeping records of the predation to see if there was more between the lights or not. And I measured different metrics of skeletal growth, keratin growth, as well as taking standardized photos of the chicks as they grow, and physiological measurements of glucose and fat. I did handheld glucometer instant readings in the field for glucose, the same process that people might use to like prick their finger, check their blood sugar. The same thing for these chicks, you just prick an artery in their wing. And fat was a visual fat score measurement. More on the standardized photos. So I took standardized photos, each with a chick laying on their belly with their chick ID, the date, and a ruler for size. And the purpose behind this was to track feather growth. So seeing, I divided the photo by like each region of the body, looking at the head, the back, the wings, the thighs, and the tail. And each body area received a threshold score, just so I can like rank and track the growth of the chicks with their feathers. And so this is all the data that I took. Let's get into some of the results. So first of all, just a general summary for the nest monitoring. There were a lot of nests in that barn. It was like about 50 just like standing nests in there. And each breeding season, there would be like between 10 to like maybe 20 pairs that nested each year. So it was really noisy, but like really cool to see that breeding colony. And overall, I found that there was no difference between clutch size, the brood size, incubation, no difference between how long it took the eggs to hatch, the incubation period, no difference between survival to the fledgling stage, and the predation events were dead equal. So no indications of differences for the nest monitoring stats. But for body mass, I did find that chicks in artificial light at night weighed less than controls by between like a gram to 1.5 grams lighter. Is a significant result there. In addition, I found that their bill lengths were shorter. Artificial light at night nestlings bill length about 0.5 millimeter to 1 millimeter shorter than control chicks. Along with keratin production, I also did some measurements of the feathers. The first graph is showing me actually measuring the feathers with the ruler and seeing the actual length of the feathers, and the tail feathers in particular were longer in light pollution than they were in control. And then the second graph is talking more about the threshold scores and showing that that the average mass that chicks start to develop their pin feathers is higher in controls than it was for light pollution. So even though the Allen nestlings kind of developed the pins at the same age, they were at a much lighter mass when they started doing that, which kind of means that the Allen nestlings were not waiting to reach like the heavier mass to put out feathers. You're just like, nope, this is the time, putting them out now. And then moving on to physiological measures of the glucose and the fat, I found no difference between blood glucose or fat between Allen and control nestlings, not significant for both, which means that like all physiological measurements were the same across the two treatments. And as well, same for skeletal growth, measuring the head length, the wing chord, and the tarsus length, those results were the same between artificial light and control nestlings, not significant. And finally, I did do a body condition index using both the wing chord for one and the tarsus length as just another metric to check. And for both of them, we found no difference in the body condition between artificial light and control. One thing that I didn't expect to see, like when planning out the methods or this experiment, was this wing asymmetry. So I saw a handful of chicks that were visually very different in the growth between the left side and the right side. This has mostly been recorded in laboratory settings or in like high stress environments, usually because of the mother's behavior during incubation. So usually the mother didn't like roll the eggs around enough so that they were like evenly baked on all sides. I would love for future studies to be able to monitor the parents along with the chicks. That was a little bit beyond the scope of this project, but obviously the parents' behavior is going to have an effect. But so since I observed this, I started measuring both sides of both the wing and the tarsus, and I subtracted the left side from the right side and ran a regression with those differences. And although I did see some visual examples, the overall results were not significant for either the wing or tarsus differences. So let's kind of sum all that up. So that was a lot of data all at once. So there was no difference in any of the nest success measures, no difference between rate of skeletal growth, physiological effects, or body condition. But what we did detect is a possible sublethal effect in the form of trade-offs between body mass and keratin-based proteins, which is evidenced by like the artificial light nestlings being lighter than the controls. In addition, we saw a trade-off between keratin allocation. Both the bill and the feathers require keratin to develop. And so we saw that the owl and nestlings had shorter bills, developed pins at a lower mass, but had longer tails. So this could indicate that there's a trade-off, like maybe they're prioritizing their feather growth over their bill growth. And so overall, the implications, artificial light might account for some of the urban phenotype. These sublethal effects are mitigated by these physiological trade-offs between mass and keratin allocation itself. These trade-offs are the reason that these effects are sublethal rather than fully lethal. We think that chicks are prioritizing their skeletal growth and metabolic maintenance at the expense of body mass and bill growth. And the reasoning behind this might be because they want to grow feathers as fast as they can to leave this stressful artificial light environment. And finally, we see all these effects, but we want to know, what do we do about it? So these are some potential insights from both my studies and summaries of other studies that have been done on nestlings and birds and light pollution. We are looking specifically at what lighting regimes might mitigate these sublethal effects. And again, I'm specifically focusing on wild birds here, although many of these solutions do apply to wildlife as well as humans. So first of all, we can look at the light intensity, which is typically measured in lux. So horizontal light emissions produce the most sky glow, and horizontal light is unenclosed bulbs that just emit light everywhere, or things like car lights or interior light escaping through uncovered windows, advertisement lighting like billboards and stadium lights. And so if the horizontal light is producing the most emissions, some things we can do to mitigate that is enclose lights downwards, like the example of the picture is. Also gonna shout out the International Dark Sky Association here. If you're interested in like sustainable lighting or more education resources related to light pollution, I highly recommend them as a source. Other options are to dim the lights, turn down their brightness, or at least flicker the lights, like maybe they're on for 2 minutes, off for 2 minutes. Even that has been found to have positive effects. And one interesting thing that I found in one study is that birds might perceive red colored lights as less intense, even if they're at the same lux. So that's really interesting thing that has to do with like the way bird eyes are structured. And the positive effects of reducing light intensity is increased melatonin secretion, which helps birds sleep, increase their immune response, reduce their stress, and on a population level, reduces building collisions, and also reduces sky glow and light trespass overall in the ecological environment. Take a sip of water real quick. The second thing to look at is the light spectra or color, which is measured in kelvins, and often called like the color temperature of the light. So overall, blue lights are more harmful than red or yellow colored lights. Widespread use of LEDs has shifted the outdoor lighting spectrum to like shorter wavelengths, which is the blue and white colored lights. And blue light has been found to scatter more strongly through the atmosphere than the longer wavelengths, which is red and yellow. So of course, the solution is to switch to the warm colored lights. As example, just you can change the color of LED lights so that you can still get the benefits of like the cost effectiveness and the low carbon emissions of LEDs, but just change the color to also benefit wildlife. And again, the positive effects in this is increased melatonin secretion, reduced stress, and less overall ecological impact. Few studies have actually, on wild songbirds specifically, have focused on the light colors. However, we can glean that overall, artificial light in any capacity has some impact on birds and wildlife in general, but at least these shorter wavelengths have less of an ecological impact, particularly less impact on birds' foraging behavior. And finally, we can adjust the light duration. Large percentage, again, of ALAN comes from street lights, advertisements, security lights, decorative lights. So it would be really beneficial to only use these types of lights when they're needed. One solution is using curfew lighting, which is just turning off or at least dimming the lights for a portion of the night. And I didn't mention it specifically in this PowerPoint slide, but studies also do show that turning off the lights has no impact on like crime rates or public safety. So positive effects for wildlife is, again, mitigate sublethal effects of artificial light. And if we dim the lights or turn off the lights during important life history events, like heavy periods of bird migration or like the start of the breeding season, that can have really important effects to facilitate like those events without harmful effects. And speaking of the curfew lighting system, I wanted to also kind of test if this would work. So I have a little bit of half-night preliminary data from the same barn swallow field experiment. I only did this in 2021. The reason it did not continue is because of the predation, and we wanted to just like increase the sample size of the full night of light treatment. So we ended up with 22 chicks and 6 nests from this artificial light setup, using the same lights that I used before, 1.6 lux warm white lights. And what I did was I turned the lights on for the first half of the night and allowed the chicks to experience natural darkness and natural photoperiod until the morning. And I used the same methods to kind of analyze this data with mixed models with random effects and repeated measures. I only compared half night to full nights. I did not include controls in this analysis. And what I found was that half night nestlings weighed more than the full night nestlings. They weighed more by like an average of 2 grams. And so they are heavier than the full night, which is really good to see. However, I also found that their bill lengths were shorter than the full night nestlings by about 1 millimeter, which isn't that big, but bill lengths for these species is like only like 7 maximum. So it could be like a huge difference. And just showing some of the other graphs and there was no difference again between half and full night in any of the physiological measurements of glucose or fat. No difference in survival or hatching, no difference in body condition. So the only significant results were the mass and the bill length. So maybe some like results and implications from that specifically. So first of all, again, no difference in physiological measures, but we found that the half-night nestlings had higher body mass and shorter bills. So what does this mean? We think that half-night nestlings might be able to allocate more protein towards their body mass because during that half-night period where the light is off, they might actually be able to get some sleep. And that sleep is really important for building proteins and putting the nutrients in the right places that they need to go. That all happens while the chick is asleep. And so they're able to build their body mass and still maintain their metabolic health. And this leads us to believe that curfew lighting could be useful for mitigating some of the sublethal effects we saw with the full night of light. And overall, that kind of concludes the in-depth work that I did. But I also wanted to know that like this research does not stop. There are many future directions that we could build upon and take this. And so I just broke a couple of them down based on like study area and the research topics. So first of all, we know that artificial light is not the only anthropogenic stressor that could be inducing an urban phenotype, although likely a significant one. But further research could explore how confounding factors like climate change and air pollution interact with and potentially worsen Allen. So atmospheric conditions like cloudy skies versus clear skies, atmospheric conditions affect the scattering of light particles. And the more particles that are in the sky, the more the light scatters. So climate-driven changes in weather conditions, especially like droughts and wildfires, we've all seen the whole like New York thing with the wildfires from Canada, that exacerbates existing air pollution and in turn might cause areas that are already heavy in Allen to appear even brighter. Another interaction between climate change and photoperiod is that the photic environment, the environment of light, might inform species range shifts. So in general, as the globe kind of warms up, species have been able to migrate further northward. However, polar species are typically adapted to extreme photic conditions, like in some cases, months of continuous light. So poleward range shifts caused by climate might be limited for organisms that can't readily adapt to longer photoperiods. And then going to like more of an organismal level, as we learned in chapter 1, the phenotypes are adaptive to natural photoperiods. So we should continue to examine how artificial light impacts phenotypic expressions, as well as the physiological mechanisms behind them, especially under different lighting colors and intensities of light. And we can also look at the markers of glucose, proteins, immune functions, and all of these different markers in response to the stress of artificial light. In particular, the ATF4 gene analysis, I think would be a very interesting next step. So ATF4 is a protein encoded by the ATF4 gene, and that protein is activated by stress. So the more stressed the bird is, the higher their ATF4 protein quantity within their bloodstream will be. So experimental expression of ATF4 might be like kind of key to understanding the genetic mechanisms that produce these observed trade-offs among energetic and morphometric morphology traits. So overall, in conclusion, understanding the relationship between types of lights and artificial lights on genotypic and phenotypic expressions will help us determine the most promising future direction of outdoor lighting that will benefit both humans and wildlife, as well as help us make more informed conservation and management decisions in urban environments. And with that, I would like to thank everyone for coming to view my dissertation. And thank some of the people without whom this research wouldn't be possible. I thank the NCSU Public Science Cluster and Southeast Climate Adaptation Science Center for funding this research. I thank my advisor, Karen, my wonderful committee, and my lab mates for helping me learn and grow as a professional. Special thanks to Charles and Cindy Snyder, as well as farm managers Zach and Nick out at Brayburn Farm, who allowed me to do research on their property and invested a lot of their time and enthusiasm into the project. I also thank all of my field technicians who came out and volunteered with me, especially Brandon and Lucy, who came out every day during some of my breeding seasons. And anyone who volunteered any amount of their time to this project, both in the field and with the photo scoring analysis. And of course, I thank my family and friends. Thank you for being here. Thank you for your constant support and prayers. And finally, I have to thank the best emotional support dog ever. He was the worst field technician, but he was still good for moral support in the end, my dog Loki. So thank you, everyone. And I think there's time for questions now.
Great, thank you, Murray. And yeah, we have a little bit of time for questions, and people can either unmute and just ask them, or you can put them in the chat, either way. And I see a lot of everyone's still giving their applause emojis.
I have a question. Do you think you will find similar results for other swallow species, like tree swallow or rough-winged swallow?
I think so. I think it would be similar for a lot of passerines, especially the migratory ones who might experience different lighting regimes as they fly to their different locations. Elsa has a question. Yes.
Thanks, Murray. Beautiful presentation. I had a question, maybe kind of along the same lines as Jen, but for the urban phenotype for birds, has that been shown in barn swallows specifically, or is that kind of a generalized thing where like little pieces of it are shown in different species? Or how much do you know about like the things that you were measuring in your birds also like occur in that same species in urban settings?
Yeah, I haven't seen any research done on barn swallows in particular showing an urban phenotype. I did kind of piece it together from like lots of other different species. The most common ones were like great tits, European blackbirds, European starlings, house sparrows. So all showing especially the signs of stress and like low immune function.
Cool. That would be interesting to like do the experiment and the observational urban gradient thing like in the same species.
Mhm.
Cool. Thanks.
Great job,
Brad.
Murray. I had a question. I mean, I'm sure on balance, light pollution is bad for birds. I was wondering if you know of any work or have any ideas as to whether there's also a world in which some version of it could be good for birds, which could maybe be confounding, like some of the reasons you didn't find as big differences. Like I was wondering if maybe, there being more ambient light later allows birds to like forage longer or something like that.
Yeah, there has been a lot of studies, especially like European studies, showing that birds do forage longer, which could help with their like nutrition or like feeding their chicks more food. There could be like an energy expense associated with that because they're not getting as much sleep, but it could be also seen as a benefit. Another one that I came across was sometimes birds increase their amount of extra pair copulations under artificial light, which increases like genetic diversity and like chance for brood survival. So some maybe positive effects.
Yeah, cool, thanks.
And there's a question in the chat.
Okay. Thanks, Aaron. Do you know if the lopsided chicks developed into lopsided adults or if that abnormality had any impact on fitness? I'm curious if you noticed anything in your system or came across any evidence in the laboratory work you mentioned reviewing. So in my study, I only saw like maybe 5 chicks across the whole 3 years of study that had that lopsided effect. And 3 of them were able to catch up enough to where they could still fledge. 2 of them got predated, so I don't know what happened. So I don't know what they would have turned into. In the laboratory evidence, a lot of it that I read about wasn't quite as severe as what I saw in my studies. So yes, the fledglings were still able to like be successful adults and like fly around. So I'm not actually quite sure what caused it to be so severe in my study system.
Hey, Murray, I have a question. Wonderful job. So my question for you is, did you find any solutions to your black rat snake problem, or did you find that it was something that you could not control?
A little bit of both. So rat snakes are very great at climbing things and squeezing into like any types of crevice on any types of surface. So there wasn't too much that I could do to control the predation, but I did manage to catch it. one time, actually 2 times. The first time I relocated it just down the street and it like came back the next day. So that wasn't enough. The second time I caught it, I drove it like further away and didn't have any more predation for the rest of the season. So yeah, it would be nice to repeat this type of study in a more controlled environment that wasn't like so exposed, like the elements and snakes. Any more questions?
Great job, Murray. I'm curious what were some of the biggest challenges for you with this project and just kind of the type of research that you undertook?
Yeah, that's a good question. I think one of the major challenges right off the bat was just the COVID-19 pandemic happening. I got to the field like late the first year because of that. I should usually start in like March to prepare for the breeding season, but I wasn't able to start until like May. So I missed a lot of the first like hatch dates and egg nest monitoring events. So just being able to overall like keep up with like the daily commute to the barn and putting up all the lights. I am terrible at like construction and like stapling things to the ceiling. So like that part was just like really intensive, but also pretty fun. So it's like the struggle was also like a great like learning opportunity and a great part of like problem solving on this project.
Murray, I have a question for you. Will this research be continued in any way? Some of your inconclusives or some of your need more research? What are the next steps for you?
I do hope to continue this. Hopefully I'll be able to repeat some of this at Mississippi State. They do have a really nice avian research center that is enclosed and able to not be predated by snakes. So maybe being able to do that and combine it with some more in-depth genetic analysis, like taking more blood samples, running more blood analysis for like proteins and genetic markers and everything. That would be my next step.
Who are some of the people that you would like to get your findings in front of? Who needs to know this information?
Definitely like urban planners, city planners, anybody who's like in charge of the lights that are put out in cities, just wanting them to know that this is a benefit for humans, this is a benefit for wildlife. You can still get all the like cost saving benefits, especially if you're turning off the lights for a period. So I hope that people who are in charge of those types of things are able to see this research and hopefully take it into account.
There's another question in the chat.
As a woman of color, did you think you had to deal with situations in the field that was hard or unexpected? So thankfully, I didn't have to deal with any like direct confrontations or any like trouble throughout most of my field seasons. It was mostly just that beginning part, looking for like field housing and kind of feeling unwelcomed, especially in like a rural area in 2020. They were like, you know, Trump flags and Confederate flags everywhere, which just made me a little bit uncomfortable, but thankfully nothing super serious happened.
Hi, Mary. I have a question. Great job, first of all. This is really awesome. I love learning about everything you've been doing. Second of all, I kind of piggybacking on the next to last question, someone had asked or what you were talking about with kind of just like actions to address this. I'm curious if you think that that is like if you've seen in the literature how that has been done, perhaps other places, or yeah, if you have recommendations or suggestions for how I don't know, like urban areas might go about enacting that. Like, do you think regulation on a city level is going to be possible, or do you think it's more just going to be like advocacy and opt-in kind of thing?
Yeah, as my role as like a researcher, I think it will be more advocacy related. Like I won't have any direct control on what lights are like put out, obviously. But I have seen a few studies that are like super regional specifics. Like in this one city, we tried dimming the lights for a period of time, and here's the results. And a lot of the studies are more focused on like measuring sky glow and the impact that like if we turn off our lights, how much does sky glow reduce. So I haven't seen too much on like these regional experimental studies with wildlife and the benefits of that, but hopefully as more and more of these studies come out, we can like begin to test things like that and hopefully apply to a broader scale.
Yeah, cool. Thank you.
All right, well, we're coming, I think that's all the questions. Unless anyone wants to jump in at the last minute here, but otherwise we're coming up on the hour. Yeah, I just want to thank everyone for coming to Murray's seminar. And yeah, it was great to see everybody, and good job, Murray. We're gonna have pretty much everyone step off, except for her committee is going to stay on, and then that's going to be like our focused like private Zoom session that'll start in a little bit.
Thank you everyone for coming.